Good morning. My name's James Stanger, and this video is asking the question, are mosaic embryos a cultural artifact, an artifact of the embryo culture system? And of course, the go-to article for anything to do with the laboratory is Jason Swain's series of articles, this one in 2019, where he has this fantastic demonstration, all these laboratory effects that can occur may impact on the embryo itself. But when you go back and look through the literature, one obvious question is, since mosaic embryos are largely a feature of the post-fertilisation events, they're mitotic rather than meiotic, does the culture conditions reflect in some way on the degree of mosaicism, and that can it be used for quality control tests? Having said that, I'd like just to pause before I go much further. So the aim of these videos is to recognise there are thousands of articles published each year on IVF and related topics. Here's IVF and here's PGT-A. And down below for this particular is the number of articles which mentions mosaic embryos. So, so the purpose of this is just to select our embryo papers, which I think are maybe important to embryologists, to IVF staff, and to their IVF clients, and to put it in some context for educational purposes. But I also like to make the point that I have got a notice board. I'm fairly optimistic, of course, and don't really expect anyone to do it. However, if you have a product or a clinic you'd like to advertise, feel free to contact FirtAid and you can put your notice up on my notice board. So enough of all this sort of rubbish. Let's get back to the topic at hand. Rates of mosaicism can vary with the testing laboratory and the interpretation of what mosaic is. In reality, it's a dog's breakfast. When they first started doing PGT-A, there was this great swag of embryos in the middle that weren't euploid, which were aneuploid, they were mosaic, and no one knew what to do with them. For a long time, they were all discarded because they were not euploid. And then some brave people started to put mosaic embryos back, and lo and behold, they got live births. And out of that came this concept that very simple mosaics, just with one segmental error, are basically normal, because what's in the intercell mass that forms a baby, excuse me, is basically euploid. So they're really euploid embryos with some mosaic trophectoderm. The ones that are more complex, would involve more than one chromosome and lots of distortions, nearly always have an abnormal intercell mass, and those embryos have been discarded. But the range is between 2 and 15%, and increasing clinics now are labelling short or simple mosaics euploid, and so we're starting to lose this concept in our literature of what is mosaic and what is a true euploid. True euploids now are euploid embryos and mosaic embryos with low levels of mosaicism. So I have to ask the question: do the degree of which cells become aneuploid in creating a mosaic embryos, is that a function of the laboratory, or is it some other inherent feature of the gamete itself? And I ask the question, therefore, if it's a laboratory thing, can that be used as a KPI tool? So, for instance, each month you'll look at the number of mosaic embryos you've biopsied, and that would be some measure of your laboratory competences. And you know what? I have not found one publication on it. If it exists, write it in the comments below and I'll find it. But I can't see any article talking using mosaic embryo rates as a KPI or quality tool. As you can see, I've done lots of videos on euploid and mosaic and aneuploid embryos, and there's more to come, of course, because this is a critical thing. However, so the next one, this video will sit just here, all right? But the reason it came to light was I was meandering around and I came across this 2018 article looking at chromosome mosaicism is impacted by compromised embryo culture conditions. And I thought, okay, so what other literature is available to reinforce the idea that the mitotic errors that occur post-fertilization are a feature of abnormal culture conditions, right? Because I've been to Jason Swain's article where he's looking at aneuploidy, and surprisingly, it's probably actually in the very early days of PGT-A, an NDS screening and mosaic definitions. So it may be a bit unfair to that paper. But in that whole paper, there's nothing that really demonstrates a strong laboratory influence on euploidy rates. So this is the one that stood out for me. Excuse me. So let's see what this says. And basically, it's a very short one. It's not open access, so I can only see the abstract, but the critical information is there. And this novel study revealed that an impact of compromised embryo culture conditions of 20% oxygen on mitotic cell divisions and chromosome segregation, resulting in post-zygotic errors and thereby elevating the expected biological representation of chromosomal mosaicism. So this, and I don't think this paper's ever really been duplicated. But they found that the overall incidence of chromosome mosaicism following biopsy of 3,000 blasts on vitrified zygotes and cultured under optimized conditions, which is 5% oxygen, was 2.3%, which is really, really low. In contrast, though, there was a significant increase in the incidence of mosaicism for human blasts cultured under compromised conditions, which is 20%. And so it's 20% oxygen gave them 20% mosaicism, and 5% oxygen gave them 2.3%, highly significant. And that's a fair number of blastocysts, 3,000 blastocysts. But once again, this is a 2018 paper, so this is right at the very early start, and it really should be replicated somewhere down the track, except no one now is doing 20% oxygen. So this resulted in the presence of two cell population, one euploid and one monosomy for a single chromosome, which typically included a small size chromosome. So in other words, the mosaicism caused by culture them in high levels of oxygen was that those are largely single chromosome segmented monosomes, a very low level of mosaicism. The remaining blasts cultured under compromised conditions of 60% of those were uniformly euploid and 20% were uniformly aneuploid. Okay. So I want to stop here because when I read this, like, this is weird, because like ever since I started, we've always grown embryos at 20% oxygen. And it's only been the last 5 or 10 years that people have decided that, in fact, 5% is okay. But people for a long time were arguing whether 5 or 20%. There's hundreds of, well, dozens of papers on it. And the reason you couldn't show was they always picked the best embryo to put back. And so you can't really make much definition of whether oxygen has an impact if you only ever put in back the best embryo, because they're always going to be implant no matter how bad the culture conditions are. But why this is important to me, because I'm a really old codger, and I started my PhD with Pat Quinn in University of Newcastle in 1977. Pat Quinn's now passed away. He was the one that gave us Quinn's human tubal media. He was a leading expert in early embryology, and I give him great respect, and I mourn his passing. Gay Harlow was a really clever researcher, very quiet, very succinct. Did 3 or 4 experiments, and they were perfect. But this is the one that, right from 1978, we knew oxygen was important. And look what she found. The optimal oxygen retention for development of pre-implantation mouse embryos at the blastocyst stage in vitro was found to be between 2.5 and 5%. In fact, other studies in humans looking at lower levels haven't really reinforced that anything less than 5% is better. 1 and 2 cell embryos had a more sharply defined range of oxygen retention capable of supporting development of the 8 cell morula. Now, remember, in those days, we only growing mouse embryos to blastocysts was a real challenge. My whole PhD was based on trying to work out fertilisation rates in mouse. This is going back to the very early start. It's amazing this paper has just been neglected for so long. It's hardly ever cited, but it's one of the landmark papers in IVF, and we should actually all put this on the wall, show how very simple, a really observant scientist can make really profound observations that take the industry 40 years to catch up. At all stages of development, more embryos develop the blastocysts in 5% compared to those in 20%. Blastocysts developing under 20% have fewer blastomeres than those developing at 5%. As the time required for development to the blastocyst stage in vitro increased, there were fewer blastomeres present at the blastocyst stage in 20% oxygen. These results indicate that the cleaving mouse embryos had an optimal oxygen requirement of about 5%. At higher oxygen tensions, fewer embryos were able to develop to the blastocyst stage, and those which do develop had fewer cell divisions. And there's 2 markers of mosaicism. One is really fast division, and the other one is really slow division. So there's an optimal rate when cells will divide. Blastomeres slowly gets faster and faster. And at that time, the mitotic separation works perfectly. But when you're too fast or too slow, it starts to fall apart. but they were more interested in cellular differentiation. So she went on and said, if a gradient of oxygen tension exists across the blastomeres from the outside of the embryo to its center, the blastomeres might be using this gradient to obtain information about their location within the embryo and respond accordingly. Thus, blastomeres on the outside at a higher oxygen tension would divide at a slower rate and form trophectoderm, whereas those on the inner cell at a lower oxygen tension would divide more rapidly and contribute to the inner cell mass. So those observant amongst my browsers will recognize that, in fact, one of the problems of mosaicism is the trophectoderm is damaged, but the inner cell mass is rarely. So even these are quite profound prophetic words written by Gay Harlow and Pat Quinn back in 1978. It was a privilege to work with them. There's hardly anything been done on this, but this other paper came from a Turkish author in 2004, I think. And they were looking at the difference between 5 and 20% in mouse embryos again. But what they could show was that the rate of inner cell mass cells to the total number was always higher with low levels of oxygen than 20% in all those cases. However, when you look at the exposure of embryos to high oxygen, that if the longer it's exposed, this is not the 3 days, there are fewer and fewer cells. But really, if you look at the stage when they're being exposed to it, it's really only the zygote stage and the early two-cell stage which are important. So the damage of oxygen is at the very start of embryo when most of the mitotic errors occur. Most people think the meiosis occurs very early in the cell division phase in the 2, 3, 4, 5 cell stage. And this is when it's most damaging. Okay. So the point I'm trying to make here is that elevated oxygen concentrations may cause an increased incidence of mosaicism, but most likely the outer cells that form the trophectoderm, and this are the cell layers that is biopsied for PGT-A, where mosaicism identified like it's a self-fulfilling prophecy. This may be why there are so many mosaic embryos still produce a healthy baby. So oxygen concentration has been now demonstrated to elevate the risk of mosaicism, but what else? I just want to say one last thing. It's interesting because I started IVF in 1982 when I finished my PhD. And from 1980, well, right from the time when Edwardson Steptoe and Alan Trounston were growing their mouse human embryos, it was always in 20% oxygen. And it wasn't until maybe 10 years ago that people started to really recognize they needed to grow at 5%. So for the first 40 years of IVF, we were growing embryos at 20% oxygen. And here there was some data that was self-evident to everyone. They just ignored those publications. That in fact they were creating mosaic embryos just because they were growing at 20% oxygen. And so for the last 40 years, 20% of all the embryos were put back and been mosaic, which probably should have been normal. They were euploid. They became mosaic because the oxygen and our miscarriage rate and our pregnancy rate have all been compromised because of that. So what's interesting, when I started looking further, so oxygen concentrate may elevate the risk of mosaicism, but what else? This was the only paper on oxygen and mosaicism. It's really interesting that there's a dearth of applications on what causes mosaicism at a environmental level, and I think there's a reason for it. There's also evidence that exposure at the early stage was more detrimental than later exposure. So I said, well, then I started looking, trying to find other publications that supported an observation that the environment may be inducing aneuploidy or mosaicism or aneuploidy in selected cells. And in this whole paper, I could be wrong. If I do, I apologize to Jason. This was the only bit of data I could show looking at the percent normal spindles against osmolarity, where if osmolarity gets really high, you have fewer and fewer mitotic spindles. So that's actually really to do with the second mitotic division. But 600 milliosmoles is really high. Now, I have to say that when you culture embryos for a long period of time, in these tiny microdrops, the osmolarity does increase from about 280 to around about 350, 340, changing the pH and becoming increasingly denser. Now, I don't know that has any effect on it, but we need to be aware that these 6 day culture of embryos to blastocysts, the microdrops are slowly becoming more and more concentrated, and there may be subtle effects there. But this paper from 2004 doesn't really give us much clues. And the only other one was looking at temperature, but this is really the minutes of each cell stage, and it doesn't give you much clue about mosaicism. It's just talking about the rates of cleavage and the rates of, basically looking at between 35 and 37. To 2 cells, it doesn't really make much difference. 3 to 4 cells, a little bit, but it isn't until you get to cavitation that the cooler temperature 35 starts to make a difference. But that information is not among mosaicism, it's just cell division. And so I said, this is really weird. There's almost as if the environment is not actually producing any mosaic embryos. That is, cell division, the embryo is not being impacted at all by embryo culture conditions. And I find that rather weird. There was a publication in 2023 looking at factors associated with embryo mosaicism, a systematic review of meta-analysis. And they showed that, in fact, the environment had almost no effect. So here's what they showed. In the present study, the authors concluded that the factors related to presence of mosaicism in embryos are the embryo biopsy day and maternal age. The rest of the study factors showed no significant relationship with mosaicism. But I don't really think they were looking at embryo culture conditions. They were looking at more patient-orientated conditions. The results are of great importance, as knowing the possible causes leading to mosaicism helps to improve the clinical results of reproductive treatments. Well, that's true. So this is their meta-analysis. I'll have to go through. Number A is embryo quality. There's no difference. Number B is delay of embryo biopsy, and there is this slight favour in terms of 5-day over day 6. Number C was maternal age, where there is clearly an effect, but it's not a great effect, just a little bit of effect, 0.92 to 0.98. D was paternal age. So in this study, there's only 3 papers, one yes, one no, and one equivocal. So this one was equivocal. This one showed that age was important, and this one showed that age was better. And C was semi-quality, but basically that's showing there's no effect at all. Then I found another paper, Factors Associated with Mosaicism in Human Embryos, a retrospective review. This study, I think they are Vietnamese authors. These findings suggest that female age is the only critical factor influencing the occurrence of mosaicism in embryos. Further research is needed to fully understand the mechanism, but I think one thing I'm going to go in future videos is looking at mitochondrial levels. And although so far I couldn't find anything linking mitochondria to mosaicism, but I suspect it could just be an energy thing. But here's this what I thought was interesting. So these are their odd ratios: female age significant, male age was significant, body mass no, AMH level no, primary infertility. Well, secondary infertility was 0.08. Now 0.08 isn't 0.05, so they said there's no difference here. But if I look at 0.08, I say, well, there's something we should look at here. So maybe something to do with secondary infertility is associated with mosaicism in some very subtle way. Looking at implantation, failure, pregnancy loss, and abnormal genetics, none of those made any difference. And also embryo quality, good or not good, that's fairly basic, isn't it? There was no difference. And biopsy day, comparing day 4, which I think is a bit, I don't know why they've done both day 4. For day 5, no difference, but they didn't actually do day 6. So obviously, you get so cross at some work. But when they went back and did a multivariate with using a generalized estimating equation system, which I don't really fully understand the stats of that, but they basically said that female age was the only important factor, and the male age was really a function of older men have older partners, generally speaking, and it's the female age which is the critical one, not the male. And then there's another one. I'm getting a bit desperate here because I can't find any other articles on the environment. So this one I thought was just interesting. This one, group of Chinese workers who do enormous amounts of genetic work, you can hardly keep up with it. They're actually saying if you look at a lot of mosaic embryos and then look at the parents, that a lot of parents have some sort of mosaicism in their germ cells. That's where the chromosomes have been swapped around to the right number. But when you put them in an embryo, you get mosaic embryos that, in fact, a lot of the mosaicism is a feature of the parental origin rather than the in vitro culture conditions. So this is almost the exact opposite to where I was going. So the prevalence of mosaicism differed significantly between PGT-A and prenatal diagnosis. There's a 12% of embryos you put back here, and what hardly any mosaic embryos when you at prenatal diagnosis. But the parental origin analysis based on pH trace, which is the genetics is kind of beyond me, too old for it really. 50% of mosaic embryos were euploid and increased the overall rate of usable embryos by 8%. So you got about another eight in every 100 embryos can be reclassified from mosaic to euploid. But the rebiopsy validation of donated embryos revealed that 94% of predicted false positive copied variant numbers were not detected in subsequent samples. Right, so almost all. This is really weird. I think, in fact, I think the industry needs to get its head around this, because when you go back and look at it, you take an embryo which has been diagnosed as mosaic and then it's not transferred, but it's rebiopsy just to validate it. In this group, 94%. I'm sure that's too high with other studies. Most of the embryos you saw were mosaic. When you retested them, they were not. Right, which goes to show the real problem is to sample a few cells and grow them up to do the testing. Part of the errors may well be occurring in the amplification phase rather than the initial cell stage. But here's the interesting thing: their paper was that 70% of the things are actually parental based and can be validated. So among mosaic embryos resulting in live birth, 66% were false positives, and only 22%, only 1/5 of all my mosaic embryos are due to mitotic errors during cell division. So it's small wonder, in fact, that you can't find any environmental effects, because in fact it's really, really low. The mitotic error rate from cells dividing, and considering this poor embryo sitting by itself, growing in culture at a really fast rate, it's amazing that only 1 in 5 of 1 in 10, almost like 1 in 20 or 1 in 30 cells will have some mitotic errors. It's actually quite profound. At least that's my interpretation of it. For embryos that are truly mosaic, our method can identify likely cause. Most of these cases are due to errors that occur after fertilisation, which are often associated with better outcomes than errors that occur before fertilisation. We won't go on to talk about the rest of the stuff. So they concluded there's no. Then I just wanted to come one last paper. I found this one. This infertility diagnosis associated with embryonic mosaicism. That is there anything to do not in maternal age, but any other feature that may be associated, that may be a marker for, or give a clue as to why there might be more mosaic embryos. And these authors, this was in 2017, just when mosaic embryos were starting to be identified, and they concluded there was no association with the cause of infertility and the rate of mosaicism in the study. Regardless, mosaicism is more frequently detected with next generation sequencing. So this is just the very opening of that technology. While some mosaic embryos lead to successful pregnancy and are eligible for transfer in circumstances, the mosaicism rate in patient population as a whole was 18%, warrants further investigation. Well, this is very early days. But their data, I think, is interesting. If you go back, they were comparing primary infertility to patients that had secondary infertility or ones that had recurrent pregnancy loss. And they were looking at that right across the board, but I think they've made a mistake. They should have just been comparing secondary to primary and recurrent pregnancy loss to primary, whereas they lumped the whole lot together. But here they're shown that age is important in mosaicism. Or this is just the age of people who have primary, secondary. So people who have secondary infertility are a bit older. They have slightly lower AMH, and they have slightly fewer eggs. Excuse me. They have roughly the same number of euploid embryos, mosaic embryos, and aneuploid embryos. But looking at when they adjust it, the euploid rate. Well, let me just go back here. Look at the mosaic rate. It's 17% for primary, 13% per embryo for secondary, and 20% for pregnant. So the secondary infertility that's caught my eye is a different group to ones that are having current pregnancy loss. So I think they made a mistake in just doing the chi-square across the difference. Aneuploid embryos, there's slightly more aneuploid in the secondary infertility loss. But look at here down here, the number of mosaic embryos, 19 for primary, 15 for secondary fertility, and 18 for pregnancy loss. And if you go back and look at that, if you do the t-test just on the means and standard deviations, the mosaic embryos, these ones here, 17%. The p-value is 0.2. But if you do, actually, I think this is, I think 19, I think I needed to, I apologize to these folks. It's this one here, aneuploid, mosaic 19 and 15. So when you adjust it per embryo, you can find that, in fact, for secondary infertility, 19 to 15 is significant. So I've sort of watched it. I'm sorry about it, but looking to me here is secondary infertility, to me, is a marker for increased levels of aneuploidy. So, and I think it's got to do with diminished ovarian reserves. People who have secondary fertility usually have a reduced ovarian reserve. They have fewer eggs. So really, what we're looking at is ovarian reserve. And what they're showing here is in this study in 2020, diminished ovarian reserve is associated with reduced euploidy rates. This one doesn't talk about mosaic, it's just euploid and aneuploidy, but you can always assume that there's 10% will be mosaic. The blastocysts from women with diminished ovarian reserve are less likely to be euploid. And this paper it was open archive, but I couldn't access it. But they could show that, in fact, looking at the people with diminished ovarian reserve, this is what the doctors have indicated. These are the euploidy rates. They start to fall when you get to 42. And this is the one with the lowest quartile of metaphase, low numbers of metaphase embryos. That also falls. And the last one was presence and types and possible factors influencing mosaicism and IVF blastocysts. This was a 21 paper. Result from a single setting, this Spanish group. Although a high interpatient variability was observed, only parental age had a positive association with mosaicism. So I'm cutting around a puff here, folks. So my comments. This video set out to examine if data demonstrated if mosaicism was a direct result of poor culture conditions. What was revealed was that most analyses report that female age was the primary and almost the only indicator of mosaicism. While aneuploid rates in other publications are higher with poorer embryo quality and lower numbers, mosaicism rates appear were not strongly linked to embryo quality. And anyway, about half or more than half the embryos of mosaic really euploid. So it seems to me here is a lot of noise about something that isn't that important. So my conclusion, dear browser, if you're still here, is that I can't really find, except for oxygen concentration, any demonstration that mosaicism rates are a feature of poor embryo quality. I'd like to stop there. My name is James Stanger, an embryologist with 50 years experience. I'm not a medical person. And this information is not medical advice, but it may help you understand what it's going on to ask your doctor what you can do. If you like what I'm doing, press subscribe or make a comment, press like. And I just like to acknowledge that this is being supported by my own company with fertilisation.com, which is an online EQA program in andrology and embryology. I just thank you for your time persevering. I know IVF is a personal journey that involves the action of many staff, many ideas, and many concepts, and it helps to understand as many of these as you can. At this point in time, I'd like to say thank you for your time and